Dear students, in this video we are going to learn about IMVIC reactions or IMVIC test. IMVIC indicates Indole Test, Methyl Red Test, Vaugus-Proskeur Test and Citrate Utilization Test. To obtain the results, 4 different tests are done. These tests are used for identification or differentiation of members of the family Enterobacteriaceae. These cultures have to be grown for 24 to 48 hours at 37 degrees Celsius and tests can be performed. So these are 4 different tests with different results when the test will be positive and the test will be negative. So these will be the standard colors obtained when the particular test is positive. The IMVIC reactions are a set of 4 useful reactions that are commonly employed in the identification of members of the family Enterobacteriaceae. These 4 tests, first test is Indole Test, second is Methyl Red Test, third is Voges-Proskauer Test and fourth is the Citrate Test. Here we can see that the red ring present which will be indicating the test is positive. Here appearance of chair ready color indicates the test is positive. In the Voges-Proskauer test, no change in the color means the test is negative. And in the citrate test, there will be appearance of Persian blue color or green color that will be indicating the test is positive. Now first we will see the Indole test. Indole test is carried out on the medium sulfide indole medium SIM or tryptophan broth or motility urease indole medium will be used, that is MIU medium. Now here the COVAX reagent is added, and if we are observing the red ring at the top, then the test will be positive, and if there is no red ring at the top, then the test is absent. So here some bacteria which are able to produce indole from amino acid tryptophan using the enzyme tryptophanase, then the tryptophan will be converted into indole and pyruvic acid along with the ammonia. Production of indole is detected using the enriched reagent or COVAX reagent. Indole reacts with the aldehyde in the reagent to give a red color. An alcoholic layer concentrates the red color as a ring at the top. So here, for obtaining this test, the bacterium to be tested is inoculated in the peptone water, which contains amino acid tryptophan, and incubated overnight at 37 degrees Celsius. Following the incubation and addition of COVAX reagent, few drops, then COVAX reagent which is containing para-dimethyl aminobenzaldehyde, isoamyl alcohol and concentrated HCl. This can be used or enriched reagent which is more sensitive to detect the indole production in aerosols or non-fermenters. Formation of a red or pink colored ring at the top is taken as positive. For example, E.Coli which shows this indole test positive while that of Klebsiella pneumonia shows the same test negative. So we can identify the E.Coli by using this test. The second test is the Methyl Red Test. This test is done to detect the ability of an organism to produce and maintain stable acid end products from glucose fermentation. Some bacteria produce large amounts of acid from glucose fermentation that they overcome by the buffering action of the system. Methyl red is a pH indicator which remains red in color at the pH of 4.4 or less. So when the bacterium is tested, is inoculated into the glucose phosphate broth which contains glucose and a phosphate buffer, and it is incubated at 37 degrees Celsius for 48 hours. Over the 48 hours, the mixed acid producing organism must produce the sufficient acid to overcome the phosphate buffer and remain acid. So the pH of the medium is tested by addition of 5 drops of methyl red reagent. Development of a red color is taken as positive, and if MR negative organism produce the yellow color instead of red color, there will be formation of yellow color which can be observed in these test tubes. One is with red color, one is with the yellow color. For example, E.Coli again, it shows the positive test of methyl red, while that of Klebsiella pneumonia shows this test negative. Third test is the Voges-Proskauer test. When we are carrying out the methyl red test, it is useful where we can have the detection of mixed acid producer. Whatever acids are produced by the bacteria, these can be detected with the help of methyl red test. So here this Vaugus-Proskauer VP test detects the butylene glycol procedures. So acetyl methyl carbinol, or commonly called as acetoin, is an intermediate in the production of butylene glycol. In this test, two reagents, that is 40% of KOH and alpha-naphthol, are added to the test broth. After incubation, it will be exposed to atmospheric oxygen. If acetoin is present, it is oxidized in the presence of air and KOH to diacetyl. Diacetyl then reacts with the gonadin components of the peptone in the presence of alpha-naphthol to produce the red color. So role of alpha-naphthol is that of the catalyst and a color intensifier. Here the bacterium will be inoculated into the glucose phosphate broth and incubated for at least 48 hours. 0.6 ml of alpha-naphthol is added to the test broth and shaken. Then 0.2 ml of 40% KOH is added to the broth and shaken. The tube is allowed to stand for 15 minutes. Appearance of a red color is taken as a positive test. The negative tubes must be held for 1 hour, since maximum color development occurs within 1 hour after the addition of reagent. So here the same reagents will be also called as Barrett's A and Barrett's B reagent. Then the fourth test is the citrate test or citrate utilization test. This particular test is performed on Simon's citrate agar. This test detects the ability of an organism to utilize citrate as the sole source of carbon and energy. Bacteria are inoculated on a medium containing sodium citrate and a pH indicator bromothymol blue. The medium also contains inorganic ammonium salts, which is utilized as a sole source of nitrogen. The dicitrate will be converted into oxaloacetate and acetate, and then further the oxaloacetate will be converted to pyruvate along with the evolution of carbon dioxide. This carbon dioxide reacts with sodium and hydrogen to produce the sodium carbonate. This utilization of the citrate involves the enzyme citratase, which breaks down citrate to oxaloacetate, and finally it is getting converted to the Na2CO3, as well as ammonia will be also utilized. Then due to the utilization of sodium citrate and ammonium salt, it results in the alkaline pH. This results in change of medium's color from green to blue. So procedure involves is the bacterial colonies are picked up from a straight wire and inoculated into the Simon's citrate agar, incubated overnight at 37 degrees Celsius. If the organism has the ability to utilize citrate, the medium changes its color from green to blue. Again here example is E. coli, this shows this test negative, and Klebsiella pneumonia shows the same test positive. Now we can see the IMVIC test of E. coli, where this is the first test, that is Indole test, where we can see the red ring at the top. So it indicates the methyl red test is, Indole test is positive. Second is the methyl red test, where we can see the red chili color at the top. So this test is positive. Then third test is the Voges-Proskauer test. There is no change color, that is yellow color is remained as it is. So this test is negative. And here at the citrate test we can see the green color remaining as it is. So it indicates that E. coli can be differentiated, which shows the Indole test and Methyl Red Test positive, while that of it doesn't shows the citrate test and the Voges-Proskauer Test. So these two tests will be negative for E. coli. It will be exactly opposite for Klebsiella pneumonia, which shows Indole test and Methyl Red Test negative, while that of Voges-Proskauer Test and Citrate Test will be positive. So here are some examples of IMVIC test for two different organisms as well. One is the protease vulgaris, which also shows the Indole test and Methyl Red Test positive and Voges-Proskauer Test and Citrate Test negative. Then comes is the Citrobacter frundi. This also shows, this is now quite different, which is showing Indole test negative, while that of it is showing Methyl Red positive. Again Voges-Proskauer negative and Citrate test positive. So these are some example of IMVIC test. Depending upon these four different tests, the bacteria can be identified or differentiated from the other bacteria by using IMVIC test, which will be involving four different tests. First test will be the Indole test, that is I stands for Indole test, M stands for Methyl Red test, V stands for Voges-Proskauer test, C stands for Citrate test. Thank you.